Welcome, Dr. Pollack. I'm really excited to have you, and I just want to introduce you. The purpose of my podcast is to introduce concepts in physiology that really are very radical or changing the dynamic of the conventional wisdom. Dr. Pollack is a scientist. He's authored over 300 research publications. He's got two award-winning books, The Fourth Phase of Water, Cells, Gels, and Engines of Life. He received the first Emoto Peace Prize and is the recipient of the University of Washington's highest honor, annual faculty lecture award. He's the founding editor-in-chief of the research journal Water and the director of the Institute for Venture Science. I could go on, but it would probably take the whole podcast. So I just want to welcome you and thank you for coming on to my podcast. And I want to start out by learning a little bit about your history. Most of your research is in water, but it's also in the physics of energy, how the body utilizes this new source of energy or this unconventional source of energy. But you were influenced by Gilbert Ling. How did you find his work? How and that seems like it really inspired you. So if you could talk about that, I'd really appreciate that.
Sure, that absolutely inspired me. I had been in a completely different field. We were interested in the molecular mechanism of muscle contraction. And I spent a couple of decades in that field. And um, it was there that I learned something about scientific sociology. Um, the person, uh, the person who had the prevailing theory of muscle contraction, Sir Andrew Huxley, a member of the Huxley family, um, master of Trinity College in Cambridge, president of the Royal Society, et cetera, et cetera. When he walked into a room, it was like hush. God had just entered, so everybody keep quiet. So among Nobel laureates, he was a laureate on top of all, you might say, the other laureates, because he had every distinction. So I learned something. We contributed greatly to the field, but few people paid attention to our uh, contributions, um, and I understood that if you're up against someone of the caliber of Sir Andrew Huxley, you're not going to win, because other people in the field, given a choice of this young upstart, I was then, and the famous godlike Sir Andrew Huxley, there's no question. So I don't have any bitterness or anything. I still maintain that we're on the right track, and his theory is, frankly, a non-starter. It disagrees with so much evidence, which people in the field ignore. So I got a sense of what it was like to fight against the mainstream. And then regarding uh, the meeting of Gilbert Ling, it happened by chance. I had no interest in water, except that muscles are mostly water.
Mhm.
um, as every tissue is mostly water. So it kind of holds some intrinsic interest. But so I went to a symposium. I was invited to a symposium to honor the memory of a famous Hungarian scientist. His name is Ernst, um, and he had two interests. One was water, and the other one was muscle contraction. So I was invited to cover muscle contraction because Ernst's ideas were not so dissimilar to mine. He knew that there was something amiss with the prevailing theory that came from Sir Andrew Huxley. Uh,
You're talking about just the actin and myosin,
yeah,
just the,
actin and
the
myosin.
ATP actin and myosin. Yeah.
Yeah,
Okay.
that's what I'm talking about. Yeah, how it actually works. I don't want
Mhm.
to get into it because it would take
Yeah.
too much time. But we had really ample evidence that the theory didn't work. and I've written about it. It appears as a chapter in my Cells, Gels, and the Engines of Life book. And I wrote a book earlier than that on the mechanism of muscle contraction. And that book, practically everybody in the field has on their shelves. But as I came to realize when I visited one of these colleagues, I saw that book, the red cover. I saw it on the bookshelf. Hey, that's my book. So he pulls it out and opens it. It had never been cracked open before. It was the first time. So I found out that, yeah, people felt obliged to purchase the book, but they didn't care about reading it. So anyway, so I went to this conference in Hungary, uh, and I gave my presentation, and I think it went okay. But then Gilbert Ling got up and gave his presentation. I was blown away. He was saying that inside the cell, and he confined his interest to inside the cell mostly. Inside the cell, the water was not the same as liquid water. So, if you have liquid water, at least according to the current understanding, the molecules are randomly disposed and bouncing around a fierce number of times per second. And he said, no, it's not like that in the cell. The molecules are organized. And he viewed the water molecules as kind of stacked or like soldiers at attention, in a row. And he said the cell was filled with that, not with liquid water. And ordinarily it would sound strange, impossible for that kind of situation. But he had a lot of evidence. And then after him came a half dozen or more independent scientists who had evidence to support his point of view. Well, as I said, I was blown away, overwhelmed, because it seemed awfully reasonable what he was talking about. But I get easily impressed by interesting ideas, and so I had to test myself. And when I returned to Seattle, I gave one of his books. By that time, he had 3 or 4. He died 5 years ago. I think there were 5 or 6. So I gave it to a few of my students who I knew could potentially have interest in the kind of subject, as a test to see what their reaction was. And the reactions were uniform. They came back to me and they said, first of all, the theory is so interesting, and if it's true, it's going to change all of biological thinking, because the pillar of thinking is that the water is liquid water, and any molecule can easily move around and diffuse in water. But he said, no, that's not the case. It's it's sort of like a crystal, like a liquid crystal. And just as a preface, when we started doing experiments, we could confirm that the water was different, as Gilbert Ling suggested, but we came on some findings that I think are well beyond Gilbert Ling, and so basic and fundamental for biology. And when you ask me the questions, I'll tell you about that. So anyway, that's how I got into it. Impressed by a lecture and other evidence that followed, and we began to do experiments. Before the experiment, it was necessary. We had no money to study water. We had money to study muscle contraction. You can't do anything in a lab without having funds. And that has turned out now to be an acute problem because our work challenges so many themes. But I decided at the time that I want to help Gilbert Ling. I want to popularize his ideas. So I wrote a book, and that was Cells, Gels, and the Engines of Life. And the first half of the book was designed to bring Gilbert Ling's ideas to the not the mainstream necessarily, but to the community, because Gilbert's writing is not so, how should I say? It doesn't read like
It's pretty deep, isn't it?
It's pretty deep.
Yeah.
It's very deep,
Yeah.
to the point where you really need practically a PhD in physical chemistry to follow every every argument he makes. And I have the feeling that the word editing never entered his lexicon. I think maybe the word doesn't exist in the Chinese language. I'm not sure. But so he'd pound something out initially on a typewriter and then the word processor, and send it off to the publisher, and the publisher would publish it. And it was so obscure that few people could really get into it, and that's not the way to market those kinds of ideas. And so I thought I was doing something noble by attempting to bring his ideas into the mainstream. Well, it turned out he hated it. He absolutely hated it, and I was shocked. But and I'm thinking, what
Why did he hate it? Why
well,
did he hate it?
his um, expressed reason was that I didn't quote him enough. And I thought I made it clear in the preface and throughout that I'm talking about Gilbert Ling's ideas, not my ideas. But he didn't see
Right.
it that way. And and one day he wrote a rather nasty email to me saying, "I don't like what you've done," and a few points, and sent it to virtually everybody in the field of water, CC. Um, it took me 10 years to um, restore our friendship. And it happened at a conference that I organized and invited him. And I got up there and presented evidence for long range ordering of water. He jumped out of his seat, and his enthusiasm was unbridled. So
Oh,
then
nice.
we made friends again. And some people found him a rather difficult guy, and he himself told me, he said, sometimes I'm at a large conference, and usually there's a ballroom and it has like 6 doors for people to come in and out, and he'd enter from one side of the room and someone else from the opposite side of the room, and when that person saw him, he made a U-turn and left because he didn't want to be associated with Gilbert Ling. Anyway, the personalities are not necessarily an issue, but I view the man as a genius, uh, and he discovered something that got us going. And so when we finally could gather some funding, we began our experiments. So I hope that answers your
Did,
question. Yeah,
yeah, did he, I haven't read much or very little of his work. Did he, I mean, a lot of his work was theoretical, but what I understand is that you actually did a lot of the experiments to validate his work. Did he do a lot of experimentation and publish work to validate his theories, or
absolutely.
was that?
No, absolutely. And his work, of course, there was some theoretical stuff, but it was mostly experimental. He did numerous experiments to test various ideas, not only the organization of water molecules, but also the role of ATP, for example. He had something different to say
Well,
about.
that is an amazing concept because most of science or all of science still to this day is so ATP focused instead of efficiency focused and energy sources like what we're going to talk about here. And he brought that into concept where, that water could be charged and create this free source of energy that we're going to talk about that you did your work for. And that's what's so revolutionary about this whole concept is to this day, every seminar you go to, it's ATP is it. That's the only source of energy we have. And so, let's get on to a little bit about your work. You were the first one to coin the phrase exclusion zone, easy water, fourth phase of water. And when you started doing this work, kind of just recap your process of discovering it and what this exclusion zone is and why it's important and how it is formed in the body, with infrared light, all the way to getting supplying the negative charge in the cell, which is fascinating because all of physiology is still based on this sodium potassium pump being maintaining this important negative charge. And what really struck me is when I went and saw you and listened to your talk, it just hit me like a brick in the head that everything is based on ATP. And it never made sense to me that 60% of the cell's energy would be used to make just to maintain a pump to keep that negative charge. It just never made sense. But I always put it kind of in the back of my mind. And when you said that, it just that is such a revolutionary paradigm shift that is not talked about. You know, I still go to seminars today. I mean, they'll talk about infrared laser or red light lasers in cytochrome C oxidase, but they never talk about how this charge is maintained. And you put it so beautifully. So if you could just go through that process
Sure.
and explain to me and ask us how that is.
Okay. Well, you ask a multitude of questions. Let's see how I can do. And if I omit answers to some of your questions, remind me, refresh me, and I'll do that. So, so Gilbert Ling was talking about the cell water as being like a liquid crystal. And, you know, crystals have a tendency, if they're pure, that if they're formed from some liquid that has contaminants, they'll push out the contaminants. Otherwise, you know, you can't get a pure crystal. And so, with that theme in mind, we were exploring to see if we could find a region, uh, and this is in a chamber, not in the cell at first, a region that excluded molecules, particles, and we succeeded first try. I don't know how lucky we're usually not so lucky. So we had a chamber. Chamber was filled with water, and we put little particles in microspheres. That was the first experiment, just tiny little spheres, 1 micrometer in diameter, which then were suspended in the water. And then we immersed in that water a small gel. Um, and we had some idea that the gel might be a good place to start. But it doesn't matter. We later substituted various polymers, biological surfaces, and such, and they give you a similar result. But the result was this: um, the particles were, or the water that surrounded the gel on all sides was particle free. We could watch the microspheres getting pushed out on all sides, and that's why we began to call it exclusion zone, because it
Mm,
included also easy is easy to remember. Exclusion zone, easy.
right.
So that made sense to us. It was a mistake because later we found we found so many features and properties of this water that had no microspheres that we began calling it fourth phase water because nothing that we measured was similar to ordinary liquid water in the subsequent measurements. So that's how it all began. And then an important, critical, I think, critically important finding was that this zone, this EZ or fourth phase water, I use those terms interchangeably. The EZ water had negative charge. Now, this was a real surprise. And what we did in the experiment, because I had some experience using the Ling Gerard, the same Ling microelectrodes, he invented them, um, where you could stick into a cell, for example, measure the electrical potential, pull it out again, and the cell would still be alive. They're kind of glass tubes that come to a sharp point, and you can stick it in, make your measurement. And so I was curious. You know, we had the lab, we had the apparatus. Why not measure? And I was actually pretty certain that we wouldn't find anything, because if you start with liquid water, which we started with, how could you wind up with a region that has negative charge? It doesn't make sense. Where does the negativity come from? And so we thought, after we found that the EZ was commonly, almost always, there's an exception, negatively charged, then there must be a region of positive charge because they have to add up to zero, right?
Right.
Um, and uh, and we found it. The region just beyond the exclusion zone, which has negative charge, has positive charge.
Which is which is the bulk water, right? What they
Bulk,
call bulk water.
right? Yeah,
Yeah.
ordinary liquid water. We call it bulk water. Yeah,
Bulk.
um, yeah. It has positive charge, and so it's a little bit of head scratching. This is like a battery. You got negative on one region, positive in another region, um, and 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 to demonstrate this, we demonstrated in multiple experiments, but the one that maybe catches your eye is we put one electrode on the negative, one electrode on the positive, and if that's like a battery, an energy source, current should flow to a load, and the load was an LED lamp, and voila, it lit the lamp. So it means it's proof of principle that you can actually extract electrical energy from
Water.
from water, which I think is pretty interesting and leads to a question of can you scale this up, which we haven't attempted yet, but that's something for the future. So the question is, well, gee, if the easy water, the creation of easy water creates a battery, where does the energy come from? You can't create something from nothing. According to one of the sort of laws of physics to which I abide, I mean this particular one, not all, um, um, that you can actually convert one. The only way you can create energy is to draw it from another form of energy. So a form of energy gets transformed, like the electric light bulb. So it's electrical energy that transforms into visible light and heat, right? And so I was wondering, we were wondering, where the hell does the energy come from? You can't take a chamber and plug it into a receptacle in the wall. So where does it come from? And I couldn't figure it out. We couldn't figure it out. It seemed rather nebulous. And then one day, it was an undergraduate. I think he was about 18 years old, fairly naive, but he was doing an experiment with the same sort of chamber that I described to you. Um, and I don't know whether either he was awfully curious or maybe bored or something, but he noticed a gooseneck lamp sitting next to him to his right. And he took the lamp and he shined it on the chamber. And the region that was illuminated, he could look at the easy, and it grew by a factor of 3 times. Impressive. So he called me in, and I went in, and I thought to myself, my goodness, eureka! This guy has found the energy. It's light energy, and I'm not so sure if he realized or realizes how profound his accidental discovery was. But so we went ahead and did more experiments, to first confirm that what he says is correct, and secondly to investigate which wavelengths of light are important. And we found we scanned through at the short end, ultraviolet, short wavelength end, the medium one, the visible spectrum, and then at long wavelength and infrared. And we found that UV had no effect, nothing noticeable. The visual spectrum, throughout the spectrum, no effect, except somewhat when we got to the reds. And then when we got to infrared, it was like gangbusters. It had a tiny amount of infrared light at 3 micrometer wavelength produced. We could get an expansion under the right conditions of like 10 times for very weak light. So we drew the conclusion that the energy comes from light, in particular infrared wavelengths, The physicists refer to that whole spectrum as light, and not just the light that we
Right.
can detect with our eyes. So yeah, so it comes from light, which in retrospect, is not unexpected because plants do exactly the same thing. They take light. Step one in the photosynthetic process is the division of water molecules into H plus and OH minus, which is essentially the same as what we found.
Same thing.
So it's not a great surprise. In fact, various considerations lead us to think that the processes are actually the same, and that photosynthesis at step one is actually uh, um, nature's most efficient way of creating everything that I've just described. Um, Now, I might interject something you didn't ask, but how is it possible that negative and positive charges can be separated? And a colleague who was in the photosynthesis field told me that, I mean, they plus and minus want to come back together. That's all they want to do. So that would annihilate any separation. And a colleague in the photosynthesis field alerted me to the fact that that issue has never been addressed by the photosynthesis people. So we've addressed it, and the answer is it appears in the fourth phase book. The answer is that the EZ is actually a matrix, a very dense matrix, and it's negatively charged. And of course, the positive charges are free in the water, and they want desperately to enter that matrix, but they can't because the matrix is so dense. And it's a it's a the structure is a series of sheets, and each sheet is hexagonal. They pile up one another, giving you a zone that is pretty large. And if you look this way, I mean, if you look at right angles to one of those sheets, you find that it's a honeycomb structure, hexagons, um, and um, and each sheet is slightly displaced from the previous one. It builds that way. So, in order for
So you're
go ahead, you got a picture of that?
so you're saying that it's actually a physical barrier to
Yes,
the positive charge,
yes, exactly.
not just electro.
Right, they
Okay,
can't get in, so they're kept separate. Yeah.
I did never realize that.
Well, I
So
think
that's a fascinating concept.
it might not have been so explicit, but I mean, it turns out to be fundamental because plus and minus don't ever want to be separated from one another. Um, that's I guess the most elementary of physical principles that we can think of. So anyway, that
So the battery doesn't dissipate then because of that physical charge until, I mean, like a battery will necessarily dissipate, but this battery, if the positive can't come in, it's basically going to hold its charge,
it
right?
does hold its charge, but like everything else, in the ideal case, of course, it should hold its charge forever. But nothing is ideal, and we find that for like a day or so. I mean, over a period of a day or 12 hours or whatever, it begins to lose its charge. And we never measured specifically the time constant of it, but it's not ideal. And, you know, some charges, I guess, do find their way in. There may be defects in that easy lattice through which charges can move slowly. I don't know. We haven't investigated that particular point, but it's not the ideal case. Yeah, in the ideal case, you're absolutely right.
So that lattice, has very unique qualities, and one of them is a semiconductor type quality. And maybe you can expound on that because I think that lattice work is key to how the that fourth phase water works. Is that correct?
Well, I'm not sure what you mean by is key. I mean, it's from what we deduced. It's a fact. Of course, we may be wrong, and then maybe it's not a fact. Maybe it's completely erroneous. But what I meant to say earlier is that that principle that you need something to prevent the recombination of all those positive charges out there, and this fits. When we came to this structure, we didn't realize that, or I didn't realize that that was an important feature. Keeping. So there needs to be a method of keeping those positive charges at bay. Otherwise, they'll just rush right in and recombine. And that's something that many people have. I mean, few people have really thought about. It's central. It's absolutely critical. So, I mean, I'm not sure. Maybe I've
Well,
forgotten your question, but
no, that's a great. And this exclusion zone water, or fourth phase water, is always next to a protein hydrophilic surface. Is that correct?
no,
You know, so that
no.
no,
Other surfaces do just fine if they're hydrophilic. It doesn't need to be a
that's
protein or a nucleic acid
okay.
surface. It could be anything, which means that we're talking about something that exists not only in the cell but all throughout nature. Whenever you have um, a hydrophilic surface, and not all of them do it, but we beginning to understand why, but most of them do, um, and you have infrared energy, um, it's going to build all over the world. It doesn't matter. And so one of the issues before you ready to jump in, maybe that's what you're going to ask me about infrared energy. Where does it come from? Um, well, were you going to ask that question? Okay,
So I mean, obviously we get it from external sources, but is it from in the body too?
it's in the body too because infrared is actually the source of heat. Um,
Right.
Infrared energy is a more fundamental um, uh, attribute than temperature. It's the infrared creates the temperature. So in your body, inside your body, you're metabolizing, and the metabolic pathways generate heat, and so that heat is available for use throughout your body. Externally, um, there are ample amounts of it. First of all, from the sun, about it was either 49 or 51% of the energy from sunlight that reaches us is in the infrared region. That's why the sun feels warm. But it's not just the sun. It's everything is that absorbs energy is radiating infrared energy out. So if you were to turn off all the lights in your room, so it's completely dark, you could see nothing, and your cell phone camera can record nothing. It's really dark. Now, if you substituted, instead of using an ordinary camera, if you substitute an infrared camera, which is pretty much the same as the ordinary camera, except the sensor is not sensitive to visible wavelengths, uh, but it's sensitive to infrared wavelengths. You get a beautiful image of everything from the microphone to the picture behind you to the doorknob. You, um, and
I
that's why
realize it's ubiquitous. Yeah.
it's
I mean
ubiquitous.
it. I mean,
Yeah,
yeah.
because if it were not generating energy that reaches the camera would record nothing. Uh, and that's why the military uses it. They use it routinely as a nightlight to check out the enemy's tanks and artillery and what have you at night, because they can see what there is. So it means that this energy is all around us, and if it's all around us, then it's available, which means that if you have the right kind of hydrophilic surface, not just proteins, yes, of course, proteins and nucleic acids, uh, but other surfaces, we've tried maybe 50 of them in different surfaces, uh, and almost all, a few exceptions, build this EZ. So in the cell, of course, there are solids in the cell, and most of the surfaces of proteins, there are some regions of the protein surface that maybe are exception to the rule, but mostly it's charged hydrophilic surfaces on the proteins. So, one idea that we have no evidence for this, but a speculation is that the protein structure is designed so that it can build this kind of water. And so um, inside the cell, it's full of nucleating surfaces that can build easy water, and the cell is filled with easy water. Uh,
Is there a reciprocal event where that energy source from this easy water is responsible for some of the protein folding, the reactions that happen with these nucleic acids, because they need an energy source, and protein folding is a essential element. So are they deriving is that process driving energy from the this process, do you think?
You ask such good questions. Yes, the answer is yes. So the second half of the book that Ling didn't like, the cells, gels, and the engines of life. The second part addressed exactly that question, and and the answer is similar to what you're alluding to. That if you have a cell with, as Gilbert Ling calls it, structured water, or we call it EZ water, or fourth phase water, it's like a crystal, and not much can happen inside of a crystal, liquid crystal. Um, Extraneous molecules can't move around because they can't get in. Um, you know, and so it's difficult to understand how the cell could work. And it occurred to me, and I did some research into this, that the cell needs to undergo some kind of transformation. And what we found and reported in that book in 5 or 6 different chapters, I investigated various tissues, and it turns out for each one of these important cell types, there's evidence that what happens is that the cell undergoes what's called a phase transition. So in the in the quiescent state, when the cell is not doing what it's designed to do, like in a muscle, it's not contracting, and a secretory cell, the cell is not secreting, etc. Um, the whole cell transforms from this kind of liquid crystal environment with proteins embedded in this. So the protein surfaces are actually creating the water, um, and um, um, so that's how should I put it? Um, that um, that's the way it actually forms. How this easy water forms. Now I've forgotten where I'm headed with this question.
Well, we're headed to eventually this negative charge is
Oh,
responsible for a lot of metabolic functions, and you had alluded to the fact that it's responsible for the negative. All cells have to maintain a negative charge, and I think you mentioned that it was like negative 60 millivolts or something like that. And and the common thought is that this negative charge was completely um, done by the sodium potassium pump. So what you described was that this negative charge that's induced by infrared light gets into the cell to maintain its negative charge. And the question I have for you is, let's say as we're aging, we're not exposed to enough infrared light. Does the sodium potassium pump, um, you know, what percentage of, in your thought, is the negative charge from the exclusion zone water versus the sodium potassium pump, and does it change as we age, and we become less metabolic, efficient? Because there's so much of our pharmaceutical industry is geared towards this pump mechanism, and they're completely missing the concept that infrared light and your concept of this negative charge, like the battery you're talking about, is responsible. So my question, well, if you could just expound on
sure.
how this negative charge how this gets into the cell, maintains it, and what role the sodium potassium pump then does play.
Sure. In short, sodium potassium pump doesn't exist. Um, there is certainly,
That's wild.
yeah, there's certainly protein there that is thought to be responsible, but there's evidence, a half dozen pieces of evidence that simply don't fit. Beyond what Gilbert Ling talked about, that so much of the energy was used to power from the cell is used to power just the sodium pump. Um, and but there are more. You could take you could take a cell and punch a large hole in the membrane, um, um, and the cell doesn't care, and um, so um, this is a process. Uh, what's it called? Where you put new DNA, electroporation. So you take the cell, you expose it to electric current. I can't remember how much electrical current, and it creates holes on the surface of the cell, or you might say the membrane of the cell. Um, and through this giant hole, obviously, small ions can pass as they like, but the cell remains perfectly normal. And the test for that is if you wait one day after you create the holes, as much as one day, you can do it, the experiment right away, but the hole has been open for one day, and you put new DNA outside, and the new DNA, big molecule, gets expressed because, the cell expresses the new protein corresponding to that DNA, which means that the DNA was able to penetrate through that opening, and everything remains absolutely normal in the cell. Another one. These are simple observations. Um, take take a raw egg. Cytoplasm is the egg white, and you you can see right away that it's a gel. It's not, or something like
Right.
that. It's not a liquid, and so and another. You can. We have done it, and others have done it. Take a gel that's similar to the inside of the cell. Uh, and stick the electrode in, one out, and you measure the same pretty much electrical potential as you do in the cell. Now, if if you can get the same result in something that looks like the cytoplasm, no membrane, no pumps, no channels, then it becomes awkward to say that the result is the result of a pump of some sort. Because if the pump is in the membrane, this has no membrane, but you get the same result. So those are just a few of the considerations, and I think that biological thinking has gone awry. There's no such thing as a pump. All the electrical potential can be attributed to the presence of negatively charged EZ water. That's
That is that is such an amazing. I cannot tell you how that should hit every scientist. And because I've been to a seminar just this year where they still, it's all sodium potassium pump. There's no mention. Every textbook to this day, there's I don't know of any textbooks that really attribute that negative charge to the process you're describing, and yet you've disproved that. But I think you alluded, and I asked you, why is science so resistant to this? And this is the whole purpose of my podcast, is because science is so resistant to change sometimes when it's so blatant and you've got proof. And I don't want to put words in your mouth, and I think it's because that's made you sort of the rebel in this process, and there's not much money in your process. But if you could expound on your thoughts on why it's so hard to get your concept accepted in the general realm right now in this day and age.
Yeah, well, it's true. In the general realm, it's ignored. On the other hand, it's very popular among people who are sort of alternative thinkers or health practitioners. The book, I guess, I have to boast a little bit. If you look on Amazon, the reviewer ratings, there are more than 700 reviewer ratings, and the score, a perfect score would be 5.0, and the score it has is 4.9. So I've never seen even the lectures of Richard Feynman, which are so popular. Our book, it has a higher score. So it is popular among some people, but not others. And the reason, I'll explain now, the reason, at least I think that so, if you start with New York Times, it was about a year ago that they addressed this question um, about revolutionary science. And they had a plot. On the x-axis was time, in year, the particular year, and on the y-axis was number of, I don't know if revolutionary is the word they use, but people who are challenging current thinking. And the graph looked like a downward slope from 1960 or something down to today. Um, and there was very little today, and they didn't really address the question of why, but they just. I think I at least I have a hypothesis why, and let me present it to you as a kind of provocative story. Suppose, which I like I like to tell. Suppose, I mean, I can look out my window from where I am now, and I can look at Lake Washington. It looks flat, and because it looks flat, I can surmise that the Earth must be flat. And everybody could see. They look at the ocean; it looks flat as far as you can see. Uh, and so the whole world thinks that the Earth is flat. But you, being a curious and inventive guy, you start thinking and you say, well, wait a second. I've seen images from satellites and I can see the curvature. And and the satellites go round and round. And I've seen pictures taken from the moon, and it looks like, the Earth is like a disc. So I don't think it's round. And you decide this issue is so critically important that you'd like to devote your life to studying it, to really check out your hypothesis. So you spend a couple of months putting together a grant proposal, being sure not to antagonize anybody, the opposition, so to speak, and you deliver it to either the National Science Foundation or NIH, National Institutes of Health, and the gatekeeper receives your application, and he says, Scott, who I don't know this guy, but I read through and I see this idea is so revolutionary that if I do my job properly, um, I'm going to recruit the most experienced and prominent reviewers to make sure, is this guy a crackpot or does he have something real? So who are these people who are recruited? They're the people you're challenging. They're the champions of the flat Earth idea. When the New York Times wants to come and investigate little bumps on the flat Earth, they're the people who are asked to comment. All right, and we all have egos. You do, I do, and we don't like to be displaced. We have ideas, and we can think that we're open scientists, but we do have those egos, some more than others, and we don't like to be dismissed. So you can imagine the reaction, the response of the reviewers. If you're right, they're wrong. They don't want to be wrong. That's exactly the way the grant system is built. And every organization I know, except one that we actually devised, Institute for Venture Science, uh, operates on that principle. You got to get the experts. And so anybody who's challenging the experts, they won't succeed. you know so I think that's the reason why what you said about there being a real dearth of genuinely revolutionary ideas. There are many ideas out there that actually don't fit the evidence, like the sodium potassium pump. By the way, there are now more than 1,000 pumps. They all need energy. So you know or putative pumps, I should say. um, yeah, okay. So I think that's the reason. The reason is the grant system. The grant system is designed in a way, inadvertently. I think some politicians must have designed it 60 or 70 years ago. Um, you got to get the experts to judge whether an application is a good one or not a good one. You need some mechanism, but it's because naturally, the ones who are chosen are the ones who are under threat by any revolutionary proposal. So the proposal won't succeed, and that's been the case. I know in my own case, I've given up. In order um, and in order for a proposal that I would write, for example, the reviewer, um, if he doesn't know or she doesn't know anything about fourth phase water, they're obliged to either read the book or somehow learn about it. And who has the time? These are busy people, and 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 so the way the system works is there has to be a threshold of real interest from several of the reviewers in order for the application to be discussed. And mine don't get to that stage. I spend a month or two preparing the application, going over every word and every phrase, and they don't even discuss it. I can't tell you how demoralizing that is. I understand the system. They're trying to save time because there are so many applications and everybody's busy. It has to change. And, well, I don't like to get into
Is
politics.
there hope?
I'm sorry?
Is there hope that it will change? Is there hope
I
that it
hope that
will change?
it changes. And, you know, um, it could change. It depends. I know if RFK Jr. Gets the position that he's been nominated for, things would definitely change. I know
Good.
views are mixed about him, and but I think
Well,
things are going.
it's got to be better than the status quo. It's
Yeah,
got to be better than
our
the
health
status
is
quo.
going downhill year after year. Where I think I read in terms of longevity, we're at number 49 this country. Imagine number 49.
Yeah, with the amount of money that we spend,
Yeah, right.
with the amount of, it's incredible when you think about it. The one thing that I see is if your ideas had a corporate product, that would probably, wake somebody up. But, and the one thing that I think your concept is possibly scalable because of this battery idea of water, and that would be a product that actually would, that concept of scalability of this battery that we're talking about is probably where money could come from. Is I don't know,
Well,
but
yeah, uh,
it's
yeah. We've of course we've thought of that. It's not just the battery concept that we have a filtration concept that, in theory, this is very preliminary, could separate salt from ocean water. Obviously these are, if they work, there's going to be major breakthroughs. But frankly, I don't want to get into any commercial enterprise. It's just not me. I'm focused on the science. The money has no appeal. Well, if someone puts $1 million in my left pocket, I'm not going to toss it away. But for me, finding truth in nature is so compelling. Uh, I don't want to do anything else. However, if other people want to take the ideas and go with them in a commercial enterprise, I'm all for it. So I welcome inquiries, and there have been some inquiries, and I think we may be getting going on some of these. And you're right. If it's a commercially useful product, a breakthrough or something, it will alert a lot of people. Um, I'm not a good
I
marketer.
just think that I just think that our research sphere is currently profit driven, product driven, corporate driven, a
Absolutely,
lot of cases.
absolutely.
And that's why the basic sciences that you are proposing revolutionary ideas don't get traction because it's not in that market of producing a product. You're talking about basic physiology 101 that should be in a textbook when people go to school and start their training. They should understand that it's not the sodium potassium pump, but this charge separation. And then that is a basis for all of these other protein folding, nucleic acids doing what they need to do. And but it's not taught because of this fundamental flaw in our system. And what's going to happen to your lab when you're not there? You're researching this water, but you've got this lab at the University of Washington. You know, can it keep going?
Well, I'm planning to live to 120. My
Okay.
naturopath told me that she's going to keep me going strong until age 120. I've got a few years left, provided I exercise enough. And this is the limitation, the exercise, because I'm so busy writing chapters and books. I've got 4 books pending, 4 of them. And
Tell me about those.
uh,
What are they?
okay, um, yeah, I didn't really answer your question yet, and
Oh, go ahead. I'm sorry, I
oh,
interrupted
okay.
you there.
What's pending? Well, the first one that's pending is the one here, and this is not the real cover. This is just a temporary placeholder. It's called Charged, and it's almost ready to send to the printer for the final version. My son is the artist, and he's a fantastic artist. And if you remember the 4th phase of Waterbook, the one that's so popular, part of the popularity is because of his artwork. It's fantastic, and he's so easy to work with. It's a delight. So that's the first one. And if you want to know something of the content, I can tell you. Um, second,
I'm just curious what you're writing.
well, let me just mention the 4 of them, and then if you want to ask us something specific, I'm more than happy to answer. The next book, which is complete and waiting for my son to start the artwork when he finishes the charge book, it's on the structure of the atom. Now, there's a structure that we all read about in the textbooks along the theme that you're suggesting, but there are flaws in in the reasoning and thinking of the model, and these are simple flaws which I can go back to and tell you that even a middle school student can understand. Nothing, and I go on to create another model, which actually is not too different from what some of the chemists of the era, they didn't like this Bohr model, the solar system kind of model with a nucleus and the electrons in orbitals around. And it turns out, I found that after I drafted the book, that one of the most prominent ones of those chemists came up with his own idea, and it turns out his idea is very similar to what I'm proposing. So I felt I I
You were validated.
validating is nice, yeah. So it was great. Okay. Anyway, that book is ready. Um, it just needs a few illustrations, and that's coming next. The next book is about volcanoes and earthquakes. Um, Now, I'm not an expert in volcanoes, even though Mount St. Helens, which blew about 40 years ago, is practically in the neighborhood in the state of Washington. And as far as earthquakes are concerned, we get them all the time, and projected sooner than rather than later is one of 9.1 magnitude. So there's a lot of interest in the Seattle area about earthquakes. So the question that I've not seen answered in a satisfactory way is, where does the energy come from? So Mount St. Helens, it used to be a beautiful mountain, cone shaped like Mount Fuji,
Yeah.
and the top quarter or so blew off. And you can imagine the amount of energy that's required, to blow it. Um, and in terms of volcanoes, somehow it's thought that heat is somehow responsible, but the idea of how heat can create something so powerful never became clear. And so I'm proposing another idea, which I think fits the evidence pretty well, about the energy, and the energy I think is the same for earthquakes and volcanoes. They're co-located for the most part. Like around the Ring of Fire, where you've got lots of volcanoes, you have earthquakes occurring in the same places. So the mechanism, therefore, is likely, not proof, likely to be the same. Anyway, that book addresses it. And then another book, which I've just half first drafted, deals with the subject that interests you the most, the idea in biology that everything you read in the textbook is not necessarily right. And what I try to bring out some of these issues is that just because it's in the textbook, you can't presume that it's necessarily right. Because the way textbooks go, if you're a publisher and you have two choices for, let's say, a physiology textbook, if they still exist, um, and one choice gives the conventional view, even if you think the conventional view is all wet, and another one gives a radical view, which one are you going to choose? Well, you're obviously going to choose the conventional one because you want to sell books. And which universities are going to subject their students, at least at the present time, to radical ideas? So they won't sell money again, and therefore they're not going to do that. So you won't easily find books written by standard publisher or published by standard venues. They won't do it. There are some things that are emerging, though. For example, I came into contact with a woman who teaches students, homeschools students, and she fell in love with fourth phase water. So she prepared a career. No, she didn't fall in love with me. She fell in love with, and so she's written a curriculum which is just about finished. I kind of helped to refine it. And it's going to be available soon for homeschooled students. And I don't know if it's going to take off or it's not going to take off, but it's necessary to expose youngsters to these ideas. Youngsters are really open to revolutionary ideas. They're not set in their ways. They want to find truth. At least most of them. So that endeavor could be an inroad into wider publicity. And also the question that you asked earlier, when I'm 120 years old and in another realm somewhere, who's going to take over? And I don't know the answer to that, but there are many people who have gone through my lab, and there are now increasing number of other laboratories who are pursuing this sort of thing. So I hope that someone will take over. And by the time I reach 120, if our lab has funding, that's the limiting factor, we will have done, I would hope, most of the essential experiments that are associated. Of course, it's never ending. Always new ideas come and they need to be tested and such. And that's what we're doing. The lab is small now. It used to be, I used to almost consistently throughout my career had 7 or 8 postdoctoral fellows, almost and undergraduate students in large numbers working in the lab. It almost became too much. And now we have many fewer because of funding issues. I was funded by a billionaire guy who found our work and tapped me on the shoulder. I'd like to fund you. Well, it wasn't exactly like that, but and he funded us for 4 years with the understanding expressed in writing that he plans to fund me indefinitely unless he runs into some unexpected financial problem. Well, he ran into a problem, and he gave me a year's notice, saying that unfortunately, most of his assets are in large complexes. I'm not sure what, large buildings or shopping centers or something like this, and they're not easy to sell. And in order to fund us, you need cash. They don't they don't want a grocery store or something like that. You need money. So he's been helping to get. And I was so thrilled to receive an email from him. He said, I'm not quoting his actual words, but he said, I've looked back on my career, And this guy was trained in physics and math. He was offered fellowships to study mathematical physics at Princeton and Berkeley and places like that, but he didn't pursue that. Um, he wrote to me saying, "I'm looking back at my career, and I feel that what I'm most proud of is supporting your work." Love,
Nice.
Peter.
That's so nice. That's so nice.
Isn't it?
That that is wonderful that people do those things because of their nature of wanting to just improve society. You
That's
know,
exactly what he
that's
was doing. Yeah, it's finding truth
uh,
is
yeah.
what he was after, and so I was pleased. So he's working to help me. We'll see if anything comes of it. Right now, we're on a shoestring.
Oh, I want to go into one more topic before we get done here, and is the idea of this structured water and memory, and what your thoughts, how memory, how this could be used for memory, and also if you intertwine any thoughts about consciousness and memory. It's a fascinating, there's a lot of debate, and I don't know, lately I've been just exploring some of the Penrose Hammerhoff theories of consciousness, but I don't know. Just if you could talk about water and memory just a little bit,
Sure.
I would love that.
Okay. Well, I start with evidence for memory, and it runs the gamut from spiritualists to Nobel laureates studying this issue. The spiritualist who most of us, many of us know about is Masaru Emoto, who passed 10 years ago. I
Hmm.
just attended the 10th anniversary memorial. I gave a talk. It was in Salzburg, Austria. Um, and for those who don't know about the topic, what he did was he took some water um, and put it on the table and projected his intention on the water. It's the main thing he did. And if he projected a positive intention like "I love you" or "You're beautiful" or something like this, and he froze the water and looked at the water that was the frozen, look at the crystals that formed, the ice crystals, they were beautiful. Um, and if he focused negative attention on it, then they were ugly. Now, um, and he wrote a book, and the book became an overnight bestseller, and millions were sold, and a lot of people were talking about it. It was 20 years ago. They produced another one after his death about 2 years ago, and I was privileged to write a foreword um, to that book. So the problem is, among scientists, almost complete rejection, and they reject it for what might be considered a good reason, because he instead of projecting his intention on one container of water, he'd do 50. And then he'd examine all 50 and he'd pick out the best one, the one that illustrated best what he wanted to show. No
Yeah,
scientist would accept that as reasonable. It's
right.
called cherry picking. And his retort was, "Well, I'm not a scientist. I'm a spiritualist, so I'm not bound by the scientific rules." So, um, but I'm not sure if you know Dean Radin, who revels in these sorts of thing, a distinguished researcher whose science is appreciated by many. Um, really objective guy. And they did a study, an independent study, and they found that in doing it the right way, that um, differences occurred, and they occurred with statistical significance, the negative versus positive intention. So, so there, it appears that there is something to it, but no scientist worth his salt will ever say, "Yeah, I like that stuff," or "It's meaningful." Okay, the second one is a guy named Jacques Benveniste, and he was a high-level French scientist, and he was working with cells called basophils, and the basophils secrete histamine, if I have it right, and they do it when the cell is specifically exposed to some antibodies. So some guy came to Jacques' lab with like 50 people in the lab and said, I can get the same result if I take those antibodies, instead of pouring the antibodies or exposing the antibodies to the cell, I can dilute it and dilute it and dilute it, as in homeopathic dilution. I could dilute it to the point where there's none of the original. All that's left is water, and get the same result. It's naturally, um, people understand that uh, it seems ridiculous. How is it possible that just ordinary water can have retain that information or memory? So he got a lot of blowback. His career was cut short by a ploy by the Nature editor, who sent several peers, including himself, to the lab to look over what they were doing. And one of those peers was a guy named uh, James Randi, who's known as um, um, what's his title? I forgot. The Amazing Randy, he goes. Well, he's passed right now. He was one of the peers, so to speak, who went to the lab. And another peer was from NIH, and he was in the Center for Scientific Integrity. In other words, he was a kind of detective to find out uh, what the tricks that some, um, you know, unscrupulous scientists might be perpetrating. So then the third one was the editor of Nature himself, and the greatest magician, who is famous for debunking the tricks of other magicians, couldn't figure out what the trick was. But they suggested sloppy science and sloppy note taking and stuff like that. And they, in the journal Nature, that's what they wrote. And from they said that water memory was a delusion. And from that, as you can imagine, it ruined the career of this important scientist, and he died prematurely. So that's the second instance. And the third one involves Nobel laureate Luc Montagnier. Montagnier was a friend of this guy, and when Jacques Benveniste died, Luc decided he got his Nobel Prize for HIV, for uh, uncovering HIV. He wanted to continue the work of Jacques Benveniste because he thought highly of Jacques, and he thought the work was worthwhile. So he came to our water conference, which I organize annually. We're up to number 17, I think, coming. And he came for a decade. He came every year and presented his results. And his results are astonishing, but confirmed in 3 other laboratories. So his experiment was, um, he took 2 vials, and both vials were factory sealed. Um, um, I mean, they couldn't. There was no possible escape of chemicals from one vial to the other vial. He put those vials in proximity of one another, and the first one contained DNA, short strand of DNA, suspended in a buffer, an aqueous buffer of water, basically. And the next one contained pure water. He held them near one another, not touching or anything, and he applied a kind of generic energy, 50 hertz or 60 hertz. And after a day or so, this one got removed, and he had this. And his hypothesis was um, that the water is storing information from the DNA, so you don't need this anymore. So he took the water that had been putatively infused with information, and he used the PCR method, the one that's used for COVID all the time, to amplify the amount of DNA. So we got the new DNA, and its sequence matched the original DNA that had been sitting near the water. So I'm not sure if you're aware of this, but um, he found it, and he presented evidence for this. at our conference. So, I mean, the bottom line, I'm just giving you a kind of capsule of the people who studied information in water. And the results are positive, ranging from a spiritualist to a Nobel laureate, and many others in between. As I said, Montagnier's work has been confirmed by two Italian groups published, and one Chinese group. That was as of a year ago. There may be even more now. I'm not sure. It's astonishing, you know. If
So, what is your theory about how water can store memory and information?
Okay, so my hypothesis, I guess, it's not risen
Of course,
to a theory
hypothesis.
yet, or maybe never will. I don't know. So um, the first one is that pure liquid water cannot store any information. The molecules are randomly disposed and they're bouncing around a fierce number of times per second or even per femtosecond. You know, they're doing this sort of thing, and so on the other hand, EZ water um, uh, has the same capability as a transistor made of silicon. Uh, let me explain that. And so it's possible that the information is stored in EZ water, and we found sometimes um, that if you put electromagnetic energy or current into water, the water transitions. It's enough to transition some of that water into EZ water. So EZ water, let me say, so if you look at a computer memory, like a memory stick or something like that, it consists of a flat layer of transistors laid out in a grid, a perfect grid, and at each point in.
I like that.
There you go. Thank you. Okay, so you can leave that up for a moment while I talk. Yeah. Okay. So each one of these blue ones is a transistor, and the transistor has two possibilities, either on or off, or zero or one, or whatever. And it's the distribution of zeros and ones throughout this array that stores the information. So the question arising is well, what about EZ water? Does it have similar properties? So the two properties that you need, one is a regular array, and the second is that the principal component of the array needs to have at least two states. Do you have a picture of the hexagonal EZ?
I think I do. Here, hold on.
Yes. Okay.
That one.
Yeah, that's exactly right. Yeah. So you see that, as I mentioned earlier, you have a substrate at the bottom, a gel or uh, polymer or biological surface, and it builds layers of EZ water. You can see 4 or 5 layers there. And if you take one layer, you can see that um, all of the hydrogens and oxygens that are in it are regularly arrayed, not rectilinear like in the transistor array, but in a regular array. And so the question arose, is it possible that either the oxygen or the hydrogen has two states? Well, it turns out it's not the hydrogen, but the oxygens contain actually not two, but 5 oxidation states. Now, this is standard chemistry. You'll see it in textbooks. They're called oxidation states or charge states. So it means that each oxygen in the array has the capability of not just two, but 5 different states. So the two conditions are satisfied, the same as occurs in your standard computer memory, except in this case, by the way, it's at the atomic scale rather than the transistor scale. And we computed that if it turns out that this is actually a substrate that could be used in computers eventually, that the computer could be the size of a pinhead because information is so dense in this. So that's where I presume that the energy is stored. And if you put it up once again, please, the same. Yeah. So you can imagine um, any one of those oxygens, which appears at the vertices. Um, you have a distribution of those, and just like the zeros and ones, um, you have the oxygens at different oxidation states, and so theoretically, it has the capacity to store information in the same way that your memory stick has that capability. And of course, we're pursuing that, and we'd like to get um, Microsoft or whatever, Intel or someone like that interested in this, because if it turns out to be valid, then it's a bonanza for one of those.
Well, it actually takes the scale even smaller than our scale that exists now.
Right,
So it
right, yeah.
and that thus allowing greater computing power and faster computing power. So do you think that as we age, that this process, as we age, we don't produce this easy water, and there's a lot of negatives, such as inflammation and lifestyle, and that is part of why memory in the brain is, as we age, not as efficient. Is your hypothesis that that this has a lot to do with memory and human brain?
Well, not only memory, but possibly loss of memory, dementia. Um,
Yeah, yeah.
I had until a few weeks ago, I had thought that dementia was irreversible. It turns out it's reversible, and the methods that are used to reverse, the ones I've heard of, are all methods used to build easy water. So it may be that as we age, our brain cells uh, become deficient in easy water. And we have evidence. I could go over it, but it's a little bit involved. Uh, that that a lot of pathology.
So just briefly, does that involve, infrared light?
Well,
What
it could.
versus
Yeah, yeah.
dementia?
The therapies. In about one of them is infrared light. Someone shared with me his experience on a patient using infrared light. The patient had dementia, and after a number of treatments, there was no more dementia. Uh, and the other one that's better documented. This was just a n equals one experience, but another one is about hyperbaric oxygen therapy. Do you know about that? You get into it.
Well, actually, I do, but I think a better way to do oxygen therapy is actually carbon dioxide therapy, and because when you increase the level of carbon dioxide, it increases the Bohr's effect, releasing the oxygen. And my problem with hyperbaric oxygen therapy is that yeah, you can get this increased oxygen temporarily, but it actually trains the hemoglobin, to hold onto the oxygen more over time. So, so I actually in my office, and personally, I've been doing my own. You can do it through breath work, holding your breath, increasing your carbon dioxide. But it does, you can actually measure tissue perfusion of oxygen by increasing your carbon dioxide, because it stimulates this Bohr's effect to release the oxygen from the hemoglobin molecule. So, yeah, you get a temporary, the hyperbaric, you get this temporary increase in oxygen. But my thinking is that in the long run, you are actually training the body to retain it. So
Okay, so that's one point of view, but uh, there's evidence that it works. My former postdoc, who knows every paper that's ever been published, he sent me 4 papers that demonstrated that the patient goes into the hyperbaric oxygen chamber, and it's reversed. So yours may be a better way to do it, but
I think that they probably both have valid. This is a guess. My guess is that long term the CO2 might be better. The short term, that the oxygen hyperbaric is better, but I don't know. It's just at
yeah,
this
I don't
point I
know
don't
either.
see.
It's not exactly my field, but we did experiments on about hyperbaric oxygen therapy. One of my students did it. What happens if you change from um, 20% oxygen to larger value, which is what happens in the hyperbaric oxygen.
Right.
Yeah. And it builds, we found, or he found, it builds easy water. And what about higher
Oh,
pressure? It also builds easy water independently, those two factors. So
mhm.
when you combine them, you're going to get a large increase or substantial increase in easy water. And if the paradigm that I'm suggesting, is correct, that if you have not enough easy water, which the cell needs to do what it does and supply energy, then the obvious therapy is to increase the amount of EZ. And both of those expedients that I told you, the infrared and the hyperbaric oxygen therapy, do exactly that. And and so I'm kind of wondering, thinking of that, if people are aware of these ideas, that you can reverse it. It's just a matter of dehydration in the brain, just the same. I mean, the easy water that fills the cell is essentially cellular
Right.
hydration, right? And
Right.
and any organ, and we have evidence for this of various organs, is deficient in EZ. It means the electrical potential, the magnitude is small. If you have not much EZ, if you have a lot of EZ, then the negative electrical potential is big. Um, and so there are,
So that's how you measure. That's how you can measure it in the brain.
well, I don't know if
With
anybody's measured it in the brain, but I'm suggesting that brain cells are, the bunch of neurons and such are not so different from kidney cells or liver cells or whatever. And in some of these tissues, I look back at evidence from 50 years ago when people were poking all kinds of cells. Cells were dysfunctional and pathological, had small electrical potential, which means not much easy water, because uh, um, you need um, those surfaces, um, uh, to create protein surfaces to build the easy water. And when you get older, there are some mutations, and not everything is the same as when you were 5 years old. Uh, and those cells, having pathological ones, having small magnitude electrical potentials, means there's not so much easy water, and therefore they can't function. So if that's the case in, say, the kidney or the liver, why might not it be the case in your brain, uh, in the neurons or whatever else exists there? So that's why I was fascinated to hear that entities or procedures that build easy water can reverse dementia.
I would love to see a study of the carbon dioxide therapy and see if that does too, because I think it theoretically makes sense, but I don't know. So anyway, well, that might be a good research project.
We've got more research projects than we can handle. I've got a long list, but until we get substantial funding, um, um, you know,
Right.
you need to pay the people who are doing the work, and so it's standard. And some people get millions from government sources, and those are mostly people who are productive and well recognized. But if you're an insurrectionist, I guess that word has been bandied around a lot. If you're an insurrectionist, um, it's really an uphill battle. I spend a good deal of my time trying to um, get people interested, but I'm no expert in marketing myself. This is a problem. Maybe.
You have a genuine essence that comes through that you are seeking knowledge for humanity's sake, and I think that's actually, you know, I think a lot of people can see that, and that's why you have so many positive reviews with your book because it comes through. And when you listen to you, it comes through, and
Well,
you
thank
can tell that you have a passion for what you're doing, which is, you know, it shows. And so I just want to compliment you on that.
Well, thank you so much. You're awfully generous, and I really appreciate your comments.
We can leave it here. I so appreciate you taking the time to do this in we're about an hour and a half in, which I think is about the attention span of most people with these podcasts. So, I could ask you a million other questions, but I just appreciate people that have the genuine curiosity to think out of the box and ask questions. And that's kind of the purpose of what I want to do with this podcast is have people like you on that ask tough questions that challenge the narrative. And maybe we're right or wrong, but the questions I think are important. And your sincerity in your books and your work is a legacy, and I just want to thank you for that. And thanks for
Well, thank you,
coming on.
Scott. Thank you for the great questions. Um, it's not always the case, but in your case, it's uh, it's really good because you've invested in the material and you understand it very well, maybe better than I do.
No, I just have more questions now. Every time you bring up something, then it's like, oh, you should do this research,
Yeah. Well,
So anyway,
frankly, I don't know how much physiology research is still. Our university they got rid of the physiology department, um, and they integrated some of the people with some genetic department or whatever. So physiology seems passe. On the other hand, it's so critically important that it's a terrible.
it's what everything is based on. It's
Yeah,
what's everything's based on physiology,
I know.
and so it's put in other curriculum. Like I'm doing a neurology fellowship program, and it's put in there, but it's not specifically. It's always just splattered in places, and so I think you're right. I'm not in the academic world anymore, and but the basic fundamentals that you're challenging are just revolutionary because everything is based on something else, and so your work showing this is fundamental to energy. I always say, really, the phenotypical expression of disease as we age can be associated with energy flux and voltage and energy gradients.
Yeah.
And so you have to look at, well, where is it where the energy coming from and what gives us efficiency? So you look at those two sides. And so if you can free up and get free energy from all these sources, and you can make yourself more efficient at delivering these energy sources, that's kind of the equation for health. And so, we don't think about that large holistic concept anymore. We reduce everything to, and I always say the definition of a specialist is somebody who knows more and more about less and less until they know everything there is to know about nothing at all, you know?
Well put.
So, so I mean, we become this reductionist idea, concept, and so we don't think of the macro picture about where is energy coming from, and how are we delivering it efficiently, and then work on those two equations.
Yep.
So,
Yep. You got it. You got it. I wish there were more people like you around. Then we would progress much more. Okay.
well, I in my little world I try to bring it, but thank you so much for all your
Thank
time.
you.
I so
Thank
appreciate it.
you. Okay. Okay. Take care. We'll see you.
Take care.
Okay. Bye bye.